This is part 2 of a 4-part video series about the molecular biology of COVID-19 in 2022.
As previously stated, the coronavirus disease is an infectious disease caused by the SARS-CoV-2 virus. One of the first things scientists did at the beginning of the coronavirus pandemic was the sequencing of the SARS-CoV-2 viral genome. This sequencing is crucial because it facilitates the characterization of the virus, which then allows scientists to infer treatment for those infected and protection in the form of vaccines to protect those at risk. Also, sequencing allows for genomic surveillance, in which the SARS-CoV-2 virus can be tracked to detect new variants, which are mutated viral genome, and monitor trends in circulating variants such as Delta or Omicron. For the detection of the SARS-CoV-2 virus, the specimen collection needs to be at the infected location, and two details are crucial in accurate results. These are the specimen type and the timing of specimen collection. Since the lungs are the most affected by this virus, the preferred specimen is a nasopharyngeal swab to collect specimen from the upper respiratory tract. Other non-invasive collection methods have been discussed, but their performance pales in comparison to the NP swabs in terms of sensitivity and specificity, which contributes to the reliability of the results. These terms respectively refer to the identification of outliers in the specimen population and the minimization of false negative results. Furthermore, the timing of specimen collection is also important in increasing the reliability of the results, in which false negative results occur more frequently when the test takes place too early or too late following exposure. For example, real-time RTPTPCR false negative rates can be minimized by using the test 2 to 3 days after the symptoms have onset, which is around 5 days post-exposure to the virus. The RTPTPCR procedure also serves as a segue into the methods of SARS-CoV-2 detection. The RTPTPCR are the preferred method for diagnostic testing due to their high degrees of sensitivity and specificity, resulting in more accurate results. The methodology for RTPTPCR follows 3 main portions. 1. The lysis and purification of viral RNA. 2. The conversion of isolated and purified RNA into DNA, and the amplification resulting in detection. To begin, the full specimen is broken apart, lysed, to access the RNA, and then purified to remove potential inhibitors for amplification. A specific primer is then added that binds to the viral RNA. If the viral RNA is not present, these primers would not bind. In the case that the viral RNA is present, the primers facilitate reverse transcriptase to convert the RNA into complementary DNA. At this point, the complementary DNA can be copied over and over again, which is amplified through the facilitation of PCR steps and in the abundance of specific primers and the Taq DNA polymerase, which eventually results in enough viral complementary DNA for detection. This is detected through the presence of fluorescent signals in which fluorescence reporter molecules increase in fluorescence as the reaction proceeds following the accumulation of the amplified viral products. Although this detection method is relatively accurate, the resource strain on its expensive required materials caused scientists to look into other potential methods, one of which is specimen pooling to increase testing capacity through combining sample pools and retesting individual samples after the identification of a positive pool. Although this would ease the burden on demand and create a more efficient procedure, it also reduces the sensitivity of the results. Another method of SARS-CoV-2 detection is through the identification of its proteins through antigen detection. This is facilitated through the lateral flow immunoassay, which is a portable and inexpensive device that only requires a sample of a few microliters. Another example where this is used is the pregnancy test. The sample is placed in a well in the device, in which the bottom of the well contains a conjugate pad that contains conjugated antibodies, which are antibodies with a molecule attached that would create a signal when the target is present. If the SARS-CoV-2 antigen is present, the conjugated antibodies would bind to it, forming a complex that moves along the device. As this occurs, the complex eventually reaches the test line to which the complex binds to binding reagents, resulting in the formation of a visibly detectable line. The PCR and antigen tests we have discussed so far both function to determine if an individual is actively infected, and each test tests for different respective aspects of the SARS-CoV-2 virus, which are genetic material and protein segments, respectively. However, we can also check to see if an individual has been priorly infected through serology tests that look for antibodies against SARS-CoV-2. An aside, antibodies are formed by the body to attack foreign substances such as the SARS-CoV-2 virus. The first antibody produced by the body when it fights a new infection is immunoglobulin M, or IgM. IgM antibodies provide short-term protection against the new infection. This is followed by IgG antibodies that can take a lengthier time to form, but are the foundation for long-term protection to prevent reinfection. IgM antibodies dwindle as time progresses post-exposure, such that they almost disappear from the blood after a few months. However, IgG antibodies can last for years, which is why some serology tests test only for IgG. Since serology tests were introduced with determining if an individual has been previously infected, you may wonder what to do if you receive a positive serology test. A positive serology test only indicates that you have the antibodies to SARS-CoV-2. Therefore, it cannot diagnose an active infection. To do so, you would need to take another test with the purpose of diagnosis.